Okay, so welcome back to this next video in which we are discussing homologous recombination to repair double-strand breaks within DNA. Okay, right, so we're just currently looking at the pathway downstream of detection of the double-strand break. Okay, and we're just about to discuss the activation of p53 protein. Okay, so we've now discussed the gene for p53. We now want to actually discuss the different domains of the actual protein. So now let's actually look at the structure of p53. Okay, so let's have a cartoon then of the structure of p53. So it's a single polypeptide, so here's the amino group. Okay, now the first important domain I'm going to draw here, okay, and this is a portion known as the transactivation domain. Okay, and we're going to see that this is the portion which binds to another protein called MDM2, okay, but we'll talk about that more in a moment. Okay, so this is the transactivation domain. Okay, right, so I'll colour this in green here. Right, then the next region after the transactivation domain is going to be a portion known as the proline-rich domain. Okay, so I'll draw it folding round down here, and this cartoon that I'm drawing is a very simplified version of how it looks in 3 dimensions. Okay, so the transactivation domain is going to be up here at the top, and then to this side we're going to have this portion known as the proline-rich domain. Okay, so called because it's very rich in the amino acid proline. Okay, so I'll just remind you of the structure of the amino acid proline, just in case you've forgotten. So, once again, we'll draw this as a residue, so we'll start by drawing the core amino acid structure. So here's the amino group. Now, in all other amino acid residues that we would draw, the amino group would have a hydrogen coming off it, but not in proline. Okay, and that's because the R group is actually going to fold back and bind to the amino group. Okay, then off the alpha carbon we have our carboxylic acid group here, which will then bind to the amino group of the next protein along. Then the R group of proline consists of 3 methylene groups, so this CH2, this means a carbon with 2 hydrogens bound off it. Okay, it looks neater if you draw the structure like this for proline. Okay, then we've got another methylene group here, another methylene group here, and then it's going to bind onto that nitrogen there. So we're forming this ring of, by adding these 3 carbons onto the side of the core amino acid structure here. And each of these 3 carbons has these 2 hydrogens also bound to it. Okay, so this is the structure of the amino acid proline within a protein, so it's a proline residue. Okay, right, so this is the proline-rich domain here. So I'll colour this proline-rich domain in turquoise here. Okay, right. Then after the proline-rich domain, it's then going to fold up like so, and then you're going to have a large portion that's going to make the DNA binding domain. Now when I draw this box, it doesn't just mean a specific portion of the linear structure. What will happen is it will, it's a huge, great mess basically here, which I'm just abbreviating to this box here. Okay, so this portion is going to make the DNA binding core domain. Okay, so this is the portion which is actually going to bind to DNA. Okay, right, so DNA binding core domain, and there should be a dash between DNA and binding. Okay, so we'll colour in the DNA binding core domain in purple, which is right at the middle of the structure of p53 here. Okay, right, and then finally, down here, okay, what we're going to have is a portion known as the tetramerization domain. Okay, and this is going to be involved in the tetramerization of p53. So in order for p53 to actually have any effect on gene expression, it firstly has to tetramerize with another 3 p53 molecules, and it's going to do this via this tetramerization domain here. Okay, so this is the tetramerization domain, and I think I'll colour in the tetramerization domain in orange. Okay, right, so what we want to discuss is what is usually stopping the activation of p53, because p53 can tetramerize, it can join together with 3 other p53 proteins, and this tetramer can act as a transcription factor which is going to change DNA expression. However, it does not usually do that, and the reason is that there are proteins which are going to bind to p53 and stop it from actually functioning. Okay, so what happens then to p53? Well, the moment that p53 is made within the cell, and we're making it all the time, all your cells are continuously making a little bit of p53. The moment it's actually made, what happens is another protein is going to come and bind to it, and it specifically binds to this transactivation domain up here. Okay, and this is MDM2 here. So MDM2 is going to bind to the transactivation domain of p53. Okay, and firstly, the binding of MDM to the transactivation domain is going to stop p53 from now functioning, but to add insult to injury, what MDM2 is also going to do is it's going to add a ubiquitin group onto p53. Okay, specifically it's going to add it onto this tetramerization domain down here. Okay, so here is a ubiquitin group being added on here. So we'll draw this, I don't think it should be in pink, we'll have it in blue. Okay, so here, this square group that's been added on to the tetramerization domain here, this is a ubiquitin group. Okay, so MDM2 is going to catalyze the addition of this ubiquitin group onto the tetramerization domain of p53. And this is not good news for p53, okay, because proteins that get ubiquitinated are going to be destroyed. Okay, so there is a formidable structure within cells, okay, known as the proteasome. Okay, and this is a truly formidable structure in the world of molecular biology. Okay, so this is basically a tube, and the idea is that proteins will go in one side of this tube, and fragments of proteins will come out the other side, basically. Okay. Now, how does a protein end up going through this tube? Well, if it gets ubiquitin added onto it, ubiquitin binds to the entrance to the tube, and then the protein will be fed through the tube of the proteasome, and as it's fed through, enzymes in the middle will just chop it into pieces, basically. It doesn't quite chop it down into amino acids, it chops it into small fragments containing a few amino acids. Okay, so basically the MDM2, by binding to the p53, firstly stops it from functioning and then has it destroyed, has it chopped into smithereens like so. Okay, so this is usually the system for making sure that p53 doesn't actually get to do anything. Okay, now, when we have got DNA damage, such as a double-strand break in our DNA, and you can't really get much more serious than a double-strand break in the DNA, what you want is you want p53 to actually become active, because p53 is going to increase the expression of genes involved in DNA repair. It's also going to increase the expression of genes that are involved in stopping the cell cycle, and also in promoting apoptosis. If the cell really can't repair its genome, you want it to commit apoptosis rather than running the risk of becoming cancerous, which is dangerous to the meta-organism, which is the organism consisting of these trillions of cells. Okay, so you want p53 activated now that we have this DNA damage, and it is the checkpoint kinase 2 and also the ataxia-telangiectasia mutated that are going to see to it that we actually do get the activation of p53. So, firstly, ataxia-telangiectasia mutated here is going to phosphorylate the MDM2 protein. Okay, so ATM is going to add a phosphate group onto the MDM2 protein, okay? And if you add a phosphate group onto the MDM2 protein, it can no longer bind to p53. Okay, so if MDM2 can't bind to p53 anymore, then it can't do this horrible thing that we've just watched happen to p53. Okay, so p53 will survive, basically. Now, the phosphate group that ATM adds on to MDM2 is specifically a phosphate group that's been added onto a serine residue, and the serine that gets this phosphate group added onto it is serine 395. Okay, so we've seen serine 139 in histone 2AX. Now we're seeing serine 395 in MDM2, which is going to be phosphorylated by ATM. Now, not only do we phosphorylate MDM2 and inactivate it, but also ATM and also checkpoint kinase 2 are both going to be involved in phosphorylating p53 to try and stop p53 from being able to bind to the MDM2. So ATM is trying to take out MDM2. It's phosphorylating MDM2 so that MDM2 can no longer bind to the transactivation domain of p53. But that's not going to take out all MDM2. We can't hope quite to do that. So we're also going to protect p53 from the MDM2 that has survived this purge. Okay? So, what are we going to phosphorylate in p53? Well, we're going to phosphorylate residues within this transactivation domain of the p53. Okay, so this first green portion here, which is, remember, is the domain which MDM2 binds to. Okay, now, ataxia-telangiectasia mutated is going to phosphorylate a serine at position 15. Okay, so serine 15 within the polypeptide of p53. Okay, checkpoint kinase 2 is going to phosphorylate a little bit further down at serine 20. Okay, so this is checkpoint kinase 2's work here. This is ataxia-telangiectasia mutated's work here. And the phosphorylation of these serines within this transactivation domain of p53 is also going to protect the p53 from being able to bind to MDM2, so it's going to stop MDM2 from being able to bind to the p53, and therefore the p53 is going to be spared this horrible fate, basically. Okay, so just to complete our picture of p53 here, remember we have these 4 other important domains here. Here is the proline-rich domain here, which once again we'll colour in turquoise. Here is the DNA binding core domain, which once again we'll colour in vivid purple here. Okay, and then we've got our tetramerization domain. Okay, then what is going to happen is once p53 survives, it's going to tetramerize, okay, so it's going to get together with 4 other p53 proteins via the binding at the tetramerization domain, and this tetramer of p53 proteins, which I'll now demote p53 just to being represented as a blob, okay, so here we have a tetramer of p53 proteins, 4 p53 proteins. This is now going to bind to DNA and change gene expression. Okay, so let me just make sure that the concept of a transcription factor is completely solid within your head. Okay, so let's let these 2 parallel lines represent a duplex of DNA, so a piece of double-stranded DNA. Okay, and let's say that this portion here is a gene. Okay, so this portion that I'm outlining, this is going to be a gene. Okay, now, upstream of genes in all eukaryotic cells, okay, and this is for every single gene, there is a region known as a promoter region. So every single gene is going to have its own promoter region in eukaryotic cells. It's not true in prokaryotic cells, but in eukaryotic cells, every single gene has its own promoter region. Now, what's the significance of this region slightly upstream of the gene? Well, the promoter region is not involved in actually being translated into protein, so it's not going to be converted into a sequence of amino acids. Okay? However, it's extremely important in controlling how much of the protein corresponding to the downstream gene that you're actually going to produce. Okay, so it's going to control the expression level of the downstream gene. Now, how does it do this? Well, basically, if you want to actually make the protein that corresponds to this gene, you need an enzyme to come and bind to the DNA. Okay, and this enzyme is RNA polymerase 2, often abbreviated to RNAP2. Okay, and RNA polymerase 2 is going to come and dock at the promoter region. It's then going to work its way down the DNA into the gene and will sequence a piece of mRNA that is complementary to the coding strand of the DNA. Okay, so RNA polymerase 2 binds to the promoter region and then works its way along the gene to produce the mRNA. So, if the promoter region has an extremely high affinity for binding to RNA polymerase 2, then RNA polymerase 2 will be binding there all the time, okay? And this means that you're going to get a huge amount of mRNA being produced that corresponds to this gene. Okay, so if RNA polymerase 2 binds there all the time, loads of mRNA is going to be produced, and that means you're going to get loads of protein. So if the promoter region is good at binding to RNA polymerase 2, that's going to lead to high expression of the gene. Whereas if it's bad at binding to the promoter region, then it will bind there hardly ever. Okay, and therefore you'll get hardly any mRNA being produced, and therefore you'll get hardly any protein being produced. Okay, so that's how promoter regions control the expression of genes. So, what then is a transcription factor? Well, a transcription factor is a molecule which is capable of binding to the promoter regions of a huge number of genes, okay? Let me just cross that out, transcription factor. Okay, so each transcription factor generally has around, well, it goes into the hundreds of genes where it will bind to the promoter region upstream of that gene. So each transcription factor has a whole plethora of promoter regions in the genome that it's capable of binding to. Okay, now when it binds to those promoter regions, at some of them it will increase the affinity of those promoter regions for binding RNA polymerase 2. Okay, therefore RNA polymerase 2 will bind to that promoter region with the transcription factor more often. It will then work its way down the downstream gene and produce mRNA more often, and therefore you'll get an increase in the production of protein because you've got an increase in the production of mRNA. Okay, at other promoter regions, this same transcription factor will decrease the affinity of RNA polymerase 2 binding to that promoter region, and therefore you'll get RNA polymerase 2 binding there less often, and therefore you'll get less mRNA being produced and therefore less protein. So each transcription factor binds to the promoter regions of a certain set of genes which it regulates, and at some it's going to increase the expression of that gene, and at others it's going to decrease the expression of that gene. So transcription factors overall are capable of changing gene expression. Some genes will have their expression increased, others will have their expression decreased. Okay, this is what this p53 tetramer is going to do. It's now going to change gene expression. It's going to increase the expression of some genes and decrease the expression of other genes. Now, for our purposes, what's important to us is that it's going to increase the expression of genes that are involved in DNA repair machinery. Okay, so it's going to increase the expression of DNA repair machinery. And you have to understand what caused this double-strand break. It was ionizing radiation. It is unlikely that the cell only got enough ionizing radiation to cause one double-strand break, it has probably hit loads of different portions of the DNA and caused loads of different types of DNA damage. So the cell is probably in the midst of a crisis here. It's probably got a huge amount of DNA damage to repair. So you're going to upregulate the proteins involved in DNA repair, basically. Okay, right. So that's now the signaling that controls how we upregulate the expression of the proteins involved in DNA repair. In the next video, what we'll turn our attention to is actually how homologous recombination is going to occur, okay, and to repair this double-strand break.